So first, welcome everyone. So today is the Poster Blitz session 3, and it's hosted by me and Haijing. She's a professor in Beijing Normal University. And I'm Fen Zhang from ICFO, Institution of Photonic Science. I'm a postdoc researcher. So now let's start the first talk. I'm going to share my screen. Can you see my screen?
Yeah.
Yes, perfect. So our first speaker is Kali Tiras. She will talk about functional brain cognitive in infancy and cognitive outcomes at 3 years. She's from Boston Children's Hospital. I'm going to give you the control. Yeah, now your floor is yours.
Great. Can everyone hear me okay? Great. Okay, thank you, Fen, so much. Yeah, so my name is Carly. This is actually my undergraduate honors thesis. I'm at Harvard at the moment, a senior, and I've been a part of the Nelson Lab at Boston Children's Hospital. So thank you guys so much for having me today. So essentially, just to give you guys an anecdote. Oh, sorry, I'm trying to switch slides. I'm not sure if I have the, oh, there we go. Okay. So just to give you guys a little anecdote, these pictures are actually me when I was around 3 years old. And there are certain signs of developing social and cognitive abilities that we can see as early as 3 years old. So there were certain experiences such as birthday parties that we're exposed to in toddlerhood to highlight how these social abilities are necessary for day-to-day functioning, like communication, forming relationships, self-regulation abilities. So for example, there are certain cognitive and social abilities that I had at 3 years old to allow me to understand that even though my favorite flavor of my birthday cake was chocolate cake, and that's the cake that I wanted at my birthday, I understood that that wasn't everyone else's favorite flavor of birthday cake. So as you can see, I have a cupcake of a chocolate cake instead of a full chocolate cake, and then I had other flavors of cake as well for other cupcakes because I understood that other kids may want strawberry or vanilla. So having these different flavors of cupcakes is kind of exemplifying my ability to understand that my desires are different from someone else's. And that's an example of something called theory of mind ability. And another example would be that my friends around me, around me blowing out my candle, they were exercising delayed gratification and self-regulation instincts by waiting to grab their favorite flavor of cupcake or not be the one to blow out my candles and have the ability to exercise that self-regulation. And that is an ability called executive functioning. So after kind of identifying these two social abilities early on in life, I next wanted to understand what brain networks would underlie these behaviors. So I used fNIRS imaging at infancy and connected that to behavioral and cognitive assessments at 3 years old. And I looked at the functional connectivity in the default mode network and frontal parietal network, which are networks that are commonly associated with social and cognitive functioning and development. And my main findings overall, I'd like to go more into it with my poster next week, but essentially we found that all the functional networks that we were looking at are significantly different from one another, and specifically they are the frontal parietal network and default mode network are significant in comparison to our control that we were looking at. And we also found that there was a negative association between the default mode connectivity and executive functioning abilities. So I'd love to go into more depth with my findings and future directions that I concluded from these results in my poster next week. I'm presenting on October 11th. So thank you guys.
Thanks, Carly. So two things I should say in the beginning. First is the seminar is recorded. Another thing is today's seminar, we don't have Q&A. So if you want to ask question, you can leave the message under the abstract of the website for each author. So now let's go to the next talk. So our next speaker is Constanza Easter. He comes from University of Genoa. He will talk about comparing motion correction techniques for resting state functional connectivity analysis in compliant healthy adults.
Thank you for the introduction. Good morning to everyone. In these few minutes, I want to introduce you to this study. I'm sorry, I'm not able to go on. Okay. As the title anticipated, in this study, the target population was a group of healthy compliant adults. They performed a resting state acquisition that lasted 15 minutes. In such kind of population, what we expect is that the presence of motion artifacts is rare. You can see an example of a channel free of motion artifact in the first row in the figure. However, when the acquisition persists for a long time, it's difficult to stay still, also for a healthy compliant adult. And so motion artifacts could arise. In the slide, you can see the plot of two different channels affected by motion artifacts. In the literature, studies approach the problem of motion artifacts in two different ways. On the one hand, some studies use a conservative performance, discarding temporal frames where motion artifacts were detected. And this approach is considered as ground truth in our work. On the other hand, some studies use motion correction. Here, we want to understand if motion correction bias the recovery of the resting state network, and we also try a good alternative to discarding motion artifacts. In fact, the discard of motion artifact prevent the calculation of some interesting parameters, such as the dynamical functional connectivity. So in our new study, we compare different pipelines to find another pipeline that performs as well as the discard one, but which maintains all temporal frames. On the left, you can see steps for each pipeline, where black arrows are the steps that all the pipeline share, while colored arrows correspond to a particular pipeline. We performed wavelet, spline, a combination of spline and wavelet, TCPA, and no correction. On the right, the figures show the percent error of the pipeline with respect to ground truth, and the results show that the best solution is to leave the motion artifacts inside the signal rather than correct them with one of these motion corrections. I would like to deepen these results with you, and so I invite you to my virtual poster on Friday from 8 to 10 a.m. And my poster ID is reported on the bottom of the slide. So I thank you for your attention, and I hope to see you soon.
Thanks, Constanza. So let's go for the next talk. The next one is, the title is Functional Human Brain Mapping in Adults with Whole Head Ultra High Density Diffuse Optical Topography. The presenter is Mohan Fogarty. Sorry. So now you should have the control.
Thank you. So thank you for the introduction and the opportunity to share my work during this Blitz session. High-density diffuse optical tomography, or HDDOT, uses a dense array of overlapping light measurements to achieve image quality close to that of fMRI. As such, the between-optoid spacing of these systems has a direct relationship on the image quality and resolution. If we were to decrease the spacing, our image resolution would increase. As shown here through simulations, with a grid spacing of 6.5 millimeters, we have a more localized point spread function compared to a spacing of 13 millimeters, resulting in a lower localization error and higher SNR. The UHD-DOT system that we developed features 255 sources by 252 detectors with an optoid spacing of 9.75 millimeters and arranged in two separate sections, resulting in approximately 10,000 measurements across two wavelengths. Next, we can assess our data quality metrics for a single high-quality subject. Data quality metrics for the system include the mean band limited SNR distributed across the cap, the light fall off to assess the optical power at source detector separations. I don't know why it's not switching. And the Fourier spectra to visualize the cardiac pulse around 1 Hertz. After assessing these data quality metrics and arriving at a system that is stable, we designed our first imaging session to focus on a variety of traditional functional neuroimaging tasks, including hearing words, generating verbs, retinotomy, and finger tapping, with a total of 17 participants completing this imaging session. Group average time traces and activation maps were computed across all subjects for each task, and beta value maps were first generated for each individual task run using a general linear model-based approach, and were then group averaged using random effects t-statistics. As a more complex task, we had 9 participants return for a second imaging session, consisting primarily of movie viewing. Participants were asked to view a 10-minute clip from The Good, the Bad, and the Ugly 2 times each. We mapped correlations between the oxyhemoglobin signal time course in each voxel and low and high level audiovisual features within the movie, such as luminance and audio envelope, allowing for the mapping of visual and auditory processing. Overall, this new system can map brain function throughout optically accessible cortex, which is promising for future studies using resting state functional connectivity, decoding, and naturalistic paradigms in healthy adults and populations that have contraindications for fMRI. And finally, this helps establish feasibility in a fiber-based system so we can continue to innovate towards wireless UHD DOT systems for a wider range of applicability for children, particularly those with cerebral palsy or autism. With that, I'd like to thank all of the co-authors and encourage you all to check out our poster during the Monday poster session, poster ID PS1 3058. Thank you.
Thanks, Cohen. So let's go to the next talk. Next one we present by Yuanyuan Gao from Boston University. Should we talk about short separation regression incorporated diffuse optical topography? So now you should have the control.
Okay, thanks for the introduction. Hello everyone, I'm Yuanyuan Gao from Boston University. So today I'm going to talk about my work in short separation regression incorporated diffuse optical tomography, SSDOT model for short. So a little bit background here is that both short separation regression and DOT image reconstruction have been shown to be effective to remove the physiological noise from fNIRS measurements. And traditionally, they are implemented sequentially. So here we hypothesize that if we do them together at the same time, can we improve the performance further? To validate our hypothesis, we designed this high-density probe, and then we use spatial and temporal basis to represent HRF changes in space domain and also time domain. And here is my modeling. So I'm not going to talk about details here, so any further detailed discussion is welcomed at my poster presentation or offline. To validate my model, I did a simulation study where I overlaid the HRFs onto the experimentally collected resting data. And then the results show that SSDOT model can improve the contrast to background ratio comparing to the sequential models. However, the improvement is marginal, especially when HRF amplitude is very small. We also did an experimental study where the participants did a box squeezing task. And then the results show that SSDOT model image reconstructed is comparable to the outcome from the sequential models, and quantitatively, SSDOT can improve CBR ratio. However, the improvement is also marginal, which is what we observed in our simulation study. So the conclusion here is that SSDOT model can improve the image quality. However, the improvement is marginal, especially when HRF amplitude is small. This also indicates the traditional sequential model already has a very strong ability to separate the brain signal from extracerebral physiological noises if we perform short separation regression and whole head DOT sequentially. That concludes my presentation. Thank you so much.
Thanks, Yuan, for the great talk. So let's welcome the next speaker. She's Caterina Amendola from Politecnico di Milano.
Thank you very much. Thank you for the presentation. Okay, so thank you for giving me the opportunity to share with you the work that we have done in collaboration with many partners, research center, companies, and hospitals. In this work, we wanted to assess the effect of red blood cell transfusion on cerebral oxygenation of preterm neonates. Indeed, we were interested in red blood cell transfusion because very often preterm neonates suffer from anemia, and the therapy for this pathology is red blood cell transfusion. Thus, 90% of extremely preterm neonates receive at least one red blood cell transfusion. However, the transfusion can lead to many complications, and very often they do not seem very effective, meaning that preterm neonates do not take any advantage from them. So it seems that the biomarker that is nowadays used to discriminate the need for red blood cell transfusion, which is the concentration of hemoglobin in the blood, is not sufficient. So in this work, we wanted to assess the effects on preterm neonates' brain hemodynamics and metabolic parameters, and we used the BabyLux device, which is a hybrid device that combines time-domain NIRS and diffuse correlation spectroscopy modules. So we performed measurements in clinics on 18 preterm neonates before and after red blood cell transfusion, and we with both our BabyLux device and a commercially available continuous wave NIRS device to assess another parameter of tissue oxygenation. Okay. So here I briefly summarize the results that we obtained. So as expected from previous studies, we've observed that red blood cell transfusion leads to an increase in the brain oxygenation of those preterm neonates, which was observed in increase of tissue oxygen saturation measured both with the BabyLux device and the INVOS device. However, with the continuous wave NIRS device, we observed a wider range of variation in the oxygenation level, suggesting that our time domain NIRS module allow us to retrieve the oxygenation of the brain in these neonates in a more precise way. And other than that, we also observed a significant improvement in the blood delivery. So an increase in total hemoglobin, oxygenated and deoxygenated hemoglobin. On the other side, however, we did not observe any significant variation in the blood flow index and in the cerebral metabolic rate of oxygen extraction that may be related to the wide range of variation of the blood flow index that was measured in different preterm neonates. So we are still working with medical doctors to understand the efficacy of the red blood cell transfusion procedure. Thank you for your attention, and if you have any question, I will be in Gatorstown in the next two hours, or either in the poster session of Monday in Boston. Thank you.
Thanks, Caterina. It was a great talk. So let's welcome our next speaker. The presenter is Clara Lunde. She's from Boston Children's Hospital. She'll talk about assessing inhibitory PEM mechanisms and neural activity in patients with endometriosis-associated PEM. The floor is yours.
Yeah, thank you for the introduction. Yeah, my name is Claire Lundy. My poster presents data that explores inhibitory pain mechanisms in people with endometriosis-associated pain. And this is data I collected in Dr. Seberg's lab at Boston Children's Hospital as part of my PhD thesis at Oxford.
Now you should have the control. Yeah.
No, it's not changing. Okay, yeah, you might have to change it for me.
Is this one? Is correct?
Yeah, but it's correct. Yeah, but it's not changing. It's not. I don't have control.
Okay, I can just help you to click the button if you need.
Okay. Yeah. So you can click again. But okay. Anyways, the endometriosis is it's a debilitating condition, incurable, and it's when the uterine tissue grows outside of the uterus. And so multiple mechanisms are thought to contribute to this associated pain, endometriosis-associated pain, specifically peripheral and central pain drivers, and which include brain and psychophysical changes. And so we tested 17 females with endometriosis and 17 pain-free controls. And these tests included measuring the magnitude of offset analgesia, which I'll refer to as OA, which has become increasingly used for assessing endogenous pain inhibition. And so an attenuated OA response has been considered to be one of the central mechanisms facilitating and contributing to pain quantification. And so OA is measured and characterized by disproportionately large reduction in pain perception after a small decrease, about 1 degree in temperature during noxious thermal stimulation. And so that's what this figure here on the left shows. That shows a 3-temperature heat stimuli that we use for OA paradigm, which was applied to the left forearm, while hemodynamic signals were measured bilaterally over the frontal and somatosensory cortices. So participants also underwent 10 minutes of resting state for hemodynamic signal measurement before the OA assessment. So they did a resting state and then the OA assessment. And so then we looked at these 6 regions of interest. So we did the resting state and OA response functional connectivity are compared using independent sample t-tests for these 6 regions. And so you can click next. And then, sorry, and so the hypothesis was that the results would show an increased functional connectivity in participants with endometriosis during both resting state and the OA testing. All right, go to the next slide. Yeah, perfect. So endometriosis participants did show an attenuated OA response, and this might suggest they have a decreased ability to modulate changes in pain perception. And then the resting state functional connectivity analysis showed that endometriosis participants decreased functional connectivity between the prefrontal and somatosensory cortices. And then for the OA functional connectivity, she showed the opposite. So endometriosis participants have increased functional connectivity between the prefrontal and somatosensory cortices as well as across hemispheres. And so the big take home from this is that about 30% of patients don't respond to our existing standard medical therapies. So you can go to the next point to standard medical therapies. And so this new approach may allow for identifying meaningful subgroups of patients who maybe process pain differently. And so maybe this could lead to more effective treatment. Yeah, thank you so much for the opportunity and listening, and hope to give more details of the study and my project at the poster.
Thanks, Class. Also, thanks for the understanding of the technical issue. So let's go for the next talk. And next talk we presented by Leanne Collins-Jones. He will talk about whole head high density diffuse optical topography in infants.
Thank you. Good morning, everyone. Or good afternoon, good evening, good night, wherever you are in the world. So as we've heard earlier in this session, high-density diffuse optical tomography, or HDDOT, is a form of fNIRS that uses a high density of channels to collect optical data in order to reconstruct an image that's localizing hemoglobin concentration changes to the cortex. And DOT has, in past, been applied to infants. So previously, the field of view of HDDOT hardware has only been capable of sampling a limited area of the cortex. And This limits our ability to investigate how brain regions across the cortical surface interact with each other. And these connectivity measures are crucial to improve our understanding of typical and atypical brain development trajectory in infants. So the aim of the work I'm presenting today was to develop headgear capable of sampling across the whole scalp surface. And if the next point wants to come up, there we go. And also to conduct a proof of concept demonstration of whole head HDDOT in infants aged 5 to 7 months. So we used the Gower Labs Lumo system to produce infant headgear, which consists of multiple independent hexagonal modules like the one we've got here, each containing 4 photodiode dual wavelength sources. So in partnership with Gower Labs, we constructed the first ever whole head HDDOT system for infant neuroimaging, consisting of 33 Lumo modules, totaling 99 sources and 132 detectors, covering a huge field of view across the cortex. So for our proof of concept study, we used 2 different screen-based paradigms: a block paradigm consisting of 3 conditions, a social condition consisting of clips of actors singing nursery rhymes, a non-social condition presenting images of modes of transport, and a baseline condition presenting modes of transport. We also used a resting state paradigm displaying non-social, non-biological moving shapes. So as for the result, from an example individual, we can see that data from channels up to 70 millimeters source detector separation pass our data quality checks for signal strength, with over 800 good channels in the range 18 to 35 millimeters. And here on the right, we also have block average responses from a single individual to the social and non-social conditions. We see greater activation in the superior temporal gyrus, temporoparietal junction bilaterally in the social condition, as we'd expect to see from previous work. And we can also see activity from across the cortical surface. These images look noisy, but I stress that this is from data from a single individual, showing we can aim to do analyses at the individual level with this hardware. So in conclusion, we've constructed the first ever whole head HDDOT system for infant neuroimaging. We're also conducting the first demonstration of whole head HDDOT in infants, and our data collection is ongoing. I'd like to acknowledge everyone who's worked on this project, my group DOT Hub at UCL, Gower Labs, the UNC-UM and Baby Connectome Project Consortium, as well as the Centre for Brain and Cognitive Development at the University of Birkbeck and the EPSRC for funding this work. I've also got 2 other posters in Boston. I'll be at every poster session, so I'd be delighted if you came to have a chat about anything. Thank you very much.
Thanks, Liam. It's a very great talk. So let's go to the next talk. It's going to be presented by Kazuma Okamoto from Chuo University. He will talk about functional connectivity during subjective assessment of audio quality in fNIRS study.
Okay, I have a little creation. Sorry. Hi, good evening. My name is Kazuma. Title of my presentation is Functional Connectivity during Subject Assessment of Audio Quality, fNIRS study. We study if brain of people can assess audio quality. The method of subject audio quality assessments is important as it can inform the choice of codec for a particular broadcast or internet service. However, the method is often affected by cognitive bias derived from listeners' experience or musical activity and their beliefs for audio quality assessments. Therefore, we focused on brain functional connectivity, by which we could present an objectively reliable evaluation method to supplement the current non-quantity method for subjective audio quality assessments. So, in this study, we examined brain functional connectivity associated with cognitive processing during subject audio quality assessments, and introduce the usefulness of a new subject audio quality assessment for listeners. Okay, next, method and analysis. 30 participants who had no experience playing instruments participated in this study. They listened to 6 musical sources which are 3 high-quality musical sources and 3 low-quality musical sources on 3 sessions like figure 1. For each evaluation session, they listen to high-quality and low-quality musical sources made from the same music. Music was classical music. High-quality audio was as good as high-resolution audio. Low-quality audio was as good as one-fifth of CD. As the pre-processing steps, we use first-degree polynomial fitting and band filtered with a range of 0.04 to 0.07 Hz, which was the difference in periodograms across each frequency at high quality and low quality. We compared functional connectivity of high quality data and low quality data by using R values, and performed paired t-tests for these subtracted z-scores between these 2 audio qualities. Okay. Next, result and discussion. We observed functional connectivity between the left superior frontal gyrus and the right medial frontal gyrus for low quality compared to high quality. The connectivity possibly related to working memory, episodic memory, and attention. Working memory refers to knowledge and experience. So in this case, working memory was observed because the low quality musical sources were different from the sound source listened on a daily basis. We were able to show listeners' brain functional connectivity during subject assessment of audio quality. If you have any question, please let's meet Gazatan or Boston. Thank you for listening.
Thanks, Kazuma. And let's go to the next talk. The next one will be talk by Wesley Becker from Children's Hospital of Philadelphia. He will talk about diffuse optical detection of intracranial hypertension in a pig-leg model of hydrocephalus. The floor is yours.
Yeah, thank you. So my poster at fNIRS will show our latest results on the optical measurement of intracranial pressure in a piglet model. See this works. Okay, sorry about that. So yeah, as you can see from this picture on the right, hydrocephalus is the accumulation of excess cerebrospinal fluid in the brain. And this increased fluid in turn can often elevate intracranial pressure to dangerously high levels that impair cerebral oxygen delivery. And it is thought that one of the causes of brain injury in this patient population is prolonged exposure to elevated ICP prior to surgical correction or treatment. So accordingly, a method that can diagnose elevated intracranial pressure at the bedside promises to save brain tissue by triggering surgical treatment prior to irreversible injury. And to this end, at the fNIRS conference last year, I presented on an optical index of intracranial pressure that could be used for this. That's derived from diffuse correlation spectroscopy measurements, cerebral blood flow pulsatility. And we did obtain encouraging data in infants to show that it works. And we're now working on extending this approach to older children with thicker skulls. So in this study, the goal is to validate the optical ICP measurement in a toddler-aged piglet model of hydrocephalus to mimic older children. And hydrocephalus is caused by the infusion of mock cerebrospinal fluid through a ventricular catheter into the brain. And we gradually increased the infusion rate to elevate ICP in increments of 5 millimeters of mercury above baseline from 5 to 60 millimeters of mercury. So we did a wide range of values. And I should say that our infusion rate was adjusted based on what the gold standard invasive ICP sensor we had in was reading. So our hypothesis was that optical intracranial pressure would correlate with the invasively measured intraparenchymal ICP. Okay, so, and encouragingly, we did see a significant correlation between the two measures. 2 of the pigs did not have usable data, but for the remaining 8 pigs, this plot is showing the optical ICP versus the invasive ICP. The most important thing I'd like to emphasize, though, is that to derive this optical ICP, we actually use this modified Beer-Lambert theory in a 2-layer head model to relate pulsatile heartbeat fluctuations to DCS changes. And that was important. If you just use the traditional semi-infinite head model, the correlation was no longer significant. And I'd love to talk with you further about this at my poster this coming Monday. And I also encourage you to check out other interesting posters from the Children's Hospital of Philadelphia from Brian, Rodrigo, and Tiffany. Thank you.
Thanks, Wesley. It was a great talk. Hope to see you in the Gathatown. So let's go to the next talk. It was presented by Alexandra Dopiarara from University of British Columbia. She will not present by Zoom, but she sent the video. So let's start it.
Yeah, the audio is not coming through.
Sorry, can you hear the sound?
No, we can't.
Let me try it again, because there's sound here, but
maybe you should share also your computer audio.
Okay. Let me check. My computer audio, yes. Can you hear it now?
Yeah, that's good.
Project that we just finished the first batch of analysis.
Thank you very much.
The idea for the project came from Dr. Lauren Emberson, the PI of her lab, watching her baby daughter play and thinking how most of what we know about what's happening inside babies' brains comes from highly structured studies with infants watching something on a screen. With the advances both in technology and in analytics, we are now in a place in our field where we can move towards more naturalistic infant fNIRS. So that's what we did. We started small. We invited 19 babies to come into the lab and play face-to-face with their parents, just as they usually do at home. We simultaneously recorded their brain and what's happening inside of the room. We had 2 main things that we were worried about. First of all, attrition is a big deal for infant neuroscience. We found that by having the parent actively engage with the baby, all of the babies were happy to play for over 5 minutes and wear the nearest cap while they do it. The second thing we were worried about was data quality. Without the structure of the task and babies being free to interact with the parent however they wanted to, we worried about motion artifacts. We run our data through the fNIRS toolbox, which allows us to see whether we can detect the heartbeat from the brain signal, and found that for 68% of babies, we had really good data quality. The second challenge that we had was how to make sense of this data. We had those rich recordings of babies interacting with the parents. And so we devised a coding scheme, which included a few key behaviors. And we had an amazing team of arrays go almost frame by frame through those recordings to code what's happening inside of the room. And for example, this is data from one baby, and you can see color-coded the behaviors that we observed, how long they lasted, and how frequently they repeated throughout the whole task. The second challenge that we had was how do we now move this data, behavioral data, and merge it with the fNIRS data that we had. We developed custom scripts that allowed us to synchronize the brain and video recordings. And then for this poster, I'm focusing just on infant directed speech, we were able to merge those behavioral codes into fNIRS data. So this graph that you see represents the raw intensity data from all of the channels, and the vertical blue lines indicate each onset of parent talking to the baby during the task. Finally, we wanted to check whether what we're seeing in the naturalistic task at least more or less corresponds to what previous studies have reported. So we've analyzed this data using a very standard approach, and we found, while we don't have any statistical analysis just yet, we found that the preliminary pilot data lets us be cautiously optimistic. We observed hemodynamic responses to infant directed speech over bilateral temporal channels, which is consistent with previous studies. While we have lots of next ideas and things to do with this rich data set, if you have any questions, come see me in Boston or catch me in Gathertown this Wednesday. Thank you so much.
Thank you, Alexandra. Sandra sent us the video. Oh, let's go for the next one. So the next one we present by Sung Ho Talk. He will talk about a deep convolution neural network for estimating hemodynamic response with reduction of motion artifacts in fNIRS. So
Hello. In this poster, I will present about a deep convolutional neural network for estimating hemodynamic response function with reduction of the motion artifact in fNIRS data. So as you may know that fNIRS signal contains the hemoglobin response to brain activation as well as noises arising from head movement and systemic physiological changes. So as you can see these figures, the motion artifact normally appears in the fNIRS measurement as spikes or baseline transitions. And this kind of artifact hinders the detection of the brain activation based on the hemodynamic measurement. So in order to address this issue, several model-based approaches have been proposed. However, some drawback is that previous methods have limitations on optimal selections of model parameters across participants or brain regions. So in order to address this issue, we applied a deep convolutional neural network to fNIRS data so that parameters of filtering and nonlinear operations can be learned from the dataset. So as a result, with minimal assumptions and tuning parameters, we achieved that hemoglobin responses can be estimated from fNIRS data with removal of the motion artifact. Sorry, the next slide cannot be moved by, yes. So this slide shows an overview of the proposed method. So proposed neural network basically based on the UNet structure, and then network parameters was trained using the semi-simulated dataset consisting of the measurements of the movement noise plus synthetic hemodynamic response functions. So here basically the input of the network consists of the measurement of the motion artifact plus synthetic hemodynamic response function, and then output of the network was set to the clean ground truth hemodynamic response functions. Then modern neural network was trained by minimizing the difference between reconstructed hemodynamic response function and the ground truth hemodynamic response functions. After training is completed, we tested the performance of this network using unseen new testing dataset. Can you move to the next slide? So this slide show, and then we compare the performance of the proposed method with the conventional approaches such as autoregressive model-based method and wavelet MDL method. So here, the dotted line is the ground truth hemodynamic response function, and the red line indicates the estimated hemodynamic response function using the proposed method. And as you can see that deep learning-based method more accurately fit the HRF compared with the other method. And this performance was further confirmed using the mean square error values. So as you can see the right side of the figure, the mean square error values of the deep learning was minimum compared to the other methods. So in conclusion, in this study, we showed that the deep learning-based method is a promising approach for estimating the hemodynamic response function with removal of the motion artifact. And thank you for your attention.
Thank you very much. So let's now go to the last talk of today. So this talk by Wan Li, and he will talk about unveiling the stimuli-evoked fNIRS response in sleep infants. The floor is yours now.
Okay, thank you, Fan. Hi everyone, I'm On from the Bionics Institute Australia. I want to share with you the poster that we'll be presenting at a conference entitled Unraveling the Stimulus-Evoked Responses in Sleeping Infants. My colleague Gautam will be at the poster session in person to share more about this study with you. So most studies reported the canonical shape hemodynamic response patterns as shown in this figure, but some studies found an inverted response. So the responses were found to vary in morphology depending on the experimental design and stimulus properties. So besides that, there is no study characterizing how the stimulus response changes over multiple stimulus epochs. So this study is designed to answer two research questions. Firstly, what is the morphology of the fNIRS response to a speech stimulus in sleeping infants? And secondly, does the response morphology change with repeated testing? So we recorded fNIRS responses from 16 sleeping infants aged between 3 to 13 months old. They were presented with a stimulus block of repeated bar speech tokens, followed by no sound period that was randomized between 22 to 32 seconds. Each participant listened to 20 trials of the bar stimulus blocks. The first result I want to share here is the HBO responses that have been averaged across every 5 trials, channels within each region of interest, and then subjects. From this zoom-in figure of the left temporal region of interest, we notice one canonical-like positive response that peaked around 6 seconds, and there was also a wide negative response that peaked around 15 seconds, and it adapts over repeated stimulus presentations. So the negative peak did not look like an undershoot because its amplitude was larger than the positive peak, and the two peak sizes were negatively correlated. So the data suggests two separate response mechanisms simultaneously evoked by an auditory stimulus. We then fitted the group average responses from trial 1 to 20 with a summation of 2 Gaussian functions that were allowed to change in peak amplitude, latency, and width independently. So this figure shows the estimated peak amplitude for each Gaussian function, and the peak of Gaussian function 1, estimated at 6 seconds, had no change of amplitude across trials. But the peak of the second Gaussian function had a drastic amplitude change from trial 1 to 10. We hypothesized 2 independent mechanisms evoked by speech stimuli in sleeping infants. If you'd like to find out more information about this study, please leave us a comment online or come chat with us during the second poster sessions. Thank you.
Oh, thanks a lot. So now we already finished all the talks today. Thanks a lot for all the speakers. You can meet up in Gathertown, and also don't forget tomorrow's virtual session in Gathertown. Bye-bye, everyone.